Hello Bio 120, welcome to week 3. This week we are going to be accomplishing one goal, and that is to obtain purified DNA from E. coli cells. Now E. coli cells are bacteria, and they have what are called plasmids as their genome. These plasmids are our DNA, the A's, T's, G's, and C's that go into everything, but they have a particular shape. They're actually a circle. And so that's something, just kind of an aside, an interesting fact about bacteria, but it's the same DNA that we have, that animals have, that everything has. And so we're going to try and be isolating that DNA today. So what we're going to do first is glove up. You will have inoculated a liquid culture of E. coli cells, and so we're just going to collect a few of those in a little 1.5 Eppendorf tube. And so what we first need to do is put our initials on that tube. We're going to be spinning them in a centrifuge. They're going to be amongst other people's samples, so you're going to want to label your tubes with your initials. Now you're going to be doing one tube. I'm going to be doing two because we need to balance the centrifuge out, and I'll explain a little bit more about that in just a little bit. So with your tubes labeled, with your Eppendorf tubes labeled, your large Eppendorf tubes, we'll fill them with cells. So you're going to pipette out about 1,500 microliters of your cell culture into your tube. Again, I'm doing two tubes. You're only going to be doing one. Then my two tubes, your one, will take over to the centrifuge, and you'll have to do this in collaboration with your lab mates. You're going to want to balance the centrifuge. There are a certain number of positions to put your microfuge tubes in, and you need the weight to be balanced across the center of that rotor that's in here. This rotor is going to spin really fast and pull down the cells that are in your culture there. So put on the screw cap tube to the screw cap top to the rotor, close that, and then put it at max speed, so speed 14, for a minute. And so what's going on in there is that rotor spinning really fast, applying a high G-force to your tubes, and those cells that are relatively heavy are going to then be pulled to the bottom of the tube, and you'll get a pellet that you probably won't be able to see in the video, but you'll have a little pellet down at the bottom of your tube when you do the lab. And that pellet is all of the cells, or the vast majority of the cells that were in that tube in suspension. Do not open the centrifuge until it is stopped. Most of them have a locking mechanism that don't release the top until they are completely stopped. And then what you should have on the bottom of your tube is a little pellet. Again, you probably can't see it in the video, but you'll have a little white pellet at the bottom of that tube. Now what we're going to do next is get rid of the liquid that's still in the tube. We want to keep the pellet but get rid of all the excess liquid. You should have a waste beaker somewhere on your bench. So that's where you will discard your supernatant, that liquid, and try to get as much of it as you can without disturbing the pellet on the bottom. The cells should stick pretty well, and so you shouldn't have to worry about it too much, but just be careful and keep an eye on your pellet as you pipette. Okay, so we've got our cells. The next thing we're going to do is treat them with a series of buffers that will gently break open these cells. Gently break them open and do so without compromising the DNA. We want intact plasmid. And so we've got MX1, MX2, and MX3. I'll refer you to your lab manual. You'll be adding them in that order, 1, 2, then 3. And I'll refer you to your lab handout as to how much of each you'll be adding. Just do that. Once you've added MX1, you're just going to pipette up and down a little bit and break up that pellet. Get all those cells off the bottom of your tube. Once you've added MX1 and resuspended the cells, you're going to add MX2. So once you've added MX2, you just want to then mix those two buffers with your cells just by inverting your tubes. So just tip them up and down a little bit. And so that MX2 is actually what's going to be lysing open your cells. And then MX3 will sort of stop that reaction. And again, once you've added MX3, just invert your tubes up and down a little bit. And again, I'm sorry, I keep saying tubes. I'm working with tubes. You're only going to have a single sample. So now we've got our lysed cells, and somewhere in here amongst protein, RNA, sugars, lipids is our DNA. To help us isolate some of that DNA, or to help clean out some of the bigger chunks of stuff that are still in those tubes, we're actually going to spin these tubes down again. This time we're going to do full speed, but we're going to do it for 5 minutes. Now, during this 5-minute spin, I'm going to just tell you about the next step. The next step is we're going to pass this mixture, the supernatant actually this time, through a column. Now, this particular column is a little plastic piece, a little white cylinder in there that you can see. Fits inside, this is the actual column part, and this is a collection tube. And so what we're going to be using is a column that fits right inside a collection tube like so. and we're going to be putting our supernatant from the tubes that are spinning right now through this column. And so what the column is going to do is that within the column there are these polymers within the column that make up a matrix. And so when we put our supernatant that's in our tubes right now through this column, all of our little circular bits of DNA are going to stick to the polymer. This polymer is going to specifically bind to our DNA. And actually what's going to happen to the rest of the stuff in our tubes is it's going to run right through. So that's going to, proteins are going to run right through, lipids are going to run right through, and sugars and other ions are going to pass right through this column, whereas the DNA is going to be trapped. It's going to be bound by this polymer. And so, in doing so, this is a really quick and easy way to isolate our DNA. And then what we're going to do is then treat this column with a wash buffer that'll help wash the rest of this junk that we don't want out of the column. Get a really nice clean column that only has DNA left in it. And then we're going to add a buffer that will actually release our plasmid from the polymer and break that bond that's going on. And then in that way, by doing the wash followed by then an elution, we can wash the other stuff away, then get a new tube and release our DNA into a new tube, and we have isolated, purified DNA. So we've got a couple of minutes left on this spin. I'm going to just cut it a little bit short for the sake of the video. Put my gloves back on. Alright, so you might see a pellet at the bottom of this of your tube, the second spin. And so as I said before, our DNA is in that liquid, is in that supernatant now. And we want that DNA, right? The whole goal of today is to isolate that DNA. So we're going to pull out the entire amount of that supernatant. Then we're going to add it to the column. Now we need to be careful. We want to add to the middle of the column. We don't want to let the liquid drop down the side of the column plastic here. And so I'll illustrate that again for you in just a second. Again, I'm doing 2 tubes. You're only going to be working with 1, but I'll have 2 tubes, 2 columns. And so what I meant by being careful to add your supernatant to the middle of the column is that if we're looking down at the tube, at our column, what you want to do is come in with your pipette tip and make sure your supernatant is dripping right into the middle of the column. And so when it hits that, it'll then soak out, and then when we spin it, most of your supernatant will run down the center of that column. You can decrease your yield of DNA if you add it to the sides and then you let that supernatant run along the outside of the column. All of the polymer is going to be or most of the polymer is going to be down the middle of this column. So we want to make sure that we try and add that, add our supernatant and containing our DNA to the middle of that column. So, okay, so we've got our columns in our collection tubes. Again, you're only working with one. And in that supernatant that we just added to it is our DNA. And so with these columns in their collection tubes, we're going to spin for just 30 seconds. So you can either push the button in this case on the centrifuge, or you can just set the dial. Making a louder noise because I forgot to screw on the top to the rotor. Not a big deal when you're doing a short spin, but I'd recommend that you put the top on. Okay, so now what you might be able to see is that the supernatant that was in our column tube has now passed through and is in our collection tube. And so that is now all of the junk, all of the stuff we don't want, the non-DNA stuff, is now in the collection tube, and our DNA is trapped in the column. So just to make sure that we've gotten all of that stuff out, you can discard what was in the collection tube from your first spin. And we're going to use now a wash buffer. And what a wash buffer is going to do is it's going to, again, wash through the column and hopefully push out any of the proteins, lipids, sugars, ions, other stuff, the non-DNA stuff, wash it all out. Because right now there might be some trapped in there. And to get really pure stuff, really pure DNA, we're going to want to wash it. So we'll have out little tubes, little Eppendorf tubes of wash buffer, and you are going to add some of that wash buffer, again, adding it to the middle of the column. Notice how I'm using two hands. You can put a hand down towards the tip of the pipette to help steady it. That's the way you want to do it. And so you'll then add or put your columns back in the centrifuge. Spin again for 30 seconds. With that wash buffer that's now passed through the column into the collection tube, you're going to discard that again into the waste. So the next thing we're going to do, we've now washed our column. Most of the other stuff, the non-DNA stuff, should be out of our column. Now what we want to do is release that DNA that's been trapped into the column. And what we do with that is we actually add an elution buffer. And an elution buffer, again, will be found in a little Eppendorf tube on your bench. Again, adding it to the middle of the column. But at this point, we're not going to leave our columns in our collection tubes. We're going to take those columns, we're actually going to add them to 1.5 Eppendorf tubes. Again, we're going to label those tubes with our initials. And then, so I will take the column out of the collection tube, and they are designed to then fit right into our Eppendorf tubes. Now the top won't close, but once we spin this, the solution buffer will pass through the column, releasing our DNA, and our DNA will now be in the bottom of an Eppendorf tube that we can close and save for later. So there's that one. Here's the other. Again, you'll only be doing one tube, one sample. You can't close the caps, but you can arrange them in such a way that you can put the top on the rotor, twist that down, and then again, spin for about 30 seconds. And at this point, the elution buffer is passing through the column, releasing all that DNA, and what we'll have in our Eppendorf tubes now is purified DNA. Okay, so it might be a little bit hard to see in the video, but we've got our sample now in our Eppendorf tubes. And then we can store this for later, for next week. So that's how you use DNA columns to isolate DNA.